I guess let's get started. I'll start with some sort of small reminder what we did yesterday, and I hope that at the moment when we start some new material, maybe some more people will join us. Okay, so we started considering the processes of metabolism and how do we get energy. So first of all, we considered what does it mean for us to get energy. We know that the ATP is basically a universal energy currency in our cells, and we know that we cannot get the ATP as it is from the environment. We should synthesize it from something else. And we have considered a way when we are synthesizing ATP from the glucose. Why? Because we eat something, that food gets digested, and we know that if we are eating some sort of sugars, that can be some complicated sugars, I don't know, some starch, some other sugars. When you eat bread, it's basically large sugar, and they get degraded to simple sugars like glucose, like fructose, and so on. And we are uptaking these simple sugars into the bloodstream, transporting them to different tissues, first of all to liver and to muscles and to brain and everywhere, and then we perform a process of getting ATP from these sort of sugars. And we have considered one of the very well-known pathways that is called the glycolysis. So here it is. Here is our glycolysis, and today I want to consider what is beyond the glycolysis. So we were able to get to the pyruvate, or maybe even to phosphoenol pyruvate, and now the idea is to consider what happens next. So how do we get from that molecule to the ATP? And we will be considering some reactions between the glycolysis and let's say cycle of reactions called Krebs cycle, or you often see TCA, tricarbon cycle, or there are some other names, but anyway, it's these sort of reactions. So we will consider these reactions, and we will still not get ATP, but we will get NADH. And with that NADH and this NADH, we will go to the electron transport chain, and I will show you how to get ATP from all that stuff. So here we are. Now I'll try to find the slide we have ended, and we will go further. Probably I'll cut some corners because it looks like I have too much material. Good. Here, that was regulation, gluconeogenesis. Yeah, so here we are. So what happens after the glycolysis? So what now? Pyruvate is some sort of a junction point. So here you see pyruvate. So here was glucose. We were able to go down with the glycolysis. We have considered glyconeogenesis, so we can go up back to the glucose. I hope you remember why do we need to resynthesize the glucose. And from here we can get to pyruvate, and well, go in different directions. So I showed you that all these metabolic pathways, they are quite convoluted, and whatever we synthesize in one metabolic pathway can be reused by many different other metabolic pathways. So here is the pyruvate. We can get with some sort of and in some sort of anaerobic conditions to lactate. We can get to alanine, or we can go here, transform that into the acetyl-CoA. That's in case of aerobic conditions, and use that acetyl-CoA either to get energy through the TCA cycle, that's Krebs cycle, or we can store that in form of fatty acids and synthesize some sort of fat. or we can get from pyruvate into, let's say, in a different pathway by synthesizing oxaloacetate and then malate or aspartate or whatever. I will not, I guess, consider all these pathways, that's a bit too much. What I want with these lectures, it's, let's say, just to show you how to analyze certain pathway so that you can make that with any other pathway you want. So if you need something, there are many pathways, and I don't know, I guess some standard biochemical courses, they consider much more of these pathways than I will do, but I just want to give you some taste of how it's performed and what kind of knowledge you can gain from considering all that articles or books on biochemistry, and if you more or less can get through these pathways, you'll be able to get, I guess, through any other, so it's not a big problem. Okay. With that being said, here is the pyruvate at junction, so we will consider these paths to acetyl-CoA, but maybe I'll tell just a few words about other possible fates for the pyruvate. So in anaerobic condition, it can be transformed into the lactate. It may sound strange for you because we are living in an atmosphere with a lot of oxygen there, but our tissues sometimes get into anaerobic conditions, especially our muscles when we need to do something very fast and we need that mechanical work right now. Our blood just cannot good enough supply them efficiently with the oxygen, so they are working for some small period of time in anaerobic conditions, and basically the pyruvate is transformed into the lactate. I will show you why do we need that. Well, actually, I showed you yesterday the Cori cycle. So when this lactate is synthesized, it can be later resynthesized in the liver, transformed in the liver back to the glucose. But there is one more reason I will show you later. It's it has to do with this NADH, NAD+. There is another form, it's the fermentation. You can transform pyruvate into acetaldehyde and then to ethanol. Well, actually you cannot, but for example, brewing yeast perfectly can do that. Well, even some animals can do that. For example, have you ever kept goldfish? No, none of you. So in anaerobic conditions, so see. Nope, no one did. Okay, so in anaerobic conditions, it turns out that tissues of goldfish are, let's say, switching to generation of ethanol. So they are basically generating that ethanol. So probably it's you can think that they are a little bit tipsy all the time. Actually, it's really easy to get rid of that ethanol if you live in water. You can just use gills to spread it away, so probably they are not swimming drunk, but anyway, it's a neat fact to know that they can synthesize ethanol in their tissues. Here is an example of ethanol fermentation in yeast. Here is pyruvate. You see that pyruvate decarboxylase using TPP and magnesium plus can get rid of this part of the molecule, so that's basically throwing away a CO2, and you get acetaldehyde. This acetaldehyde, with the help of alcohol dehydrogenase, can be transformed into ethanol. So here it is. So you basically transform this oxygen into OH, and you get this H from the NADH plus H+. So you use NADH, you get NAD+ at the end of the day, and this oxygen is transformed into OH. Okay, what else? That's a small reminder that during anaerobic metabolism, lactate is produced by muscle tissue and released into the bloodstream. So here is once again the Cori cycle, and here's how that works. So I'm not getting into these details. but I will show you this thing. So we have an enzyme called lactate dehydrogenase, and you see that it can transform pyruvate into the lactate. So here how they are different. Oops. So once again we transform oxygen into the OH group. Here are some sort of 3D view and these for this sort of enzyme. I will not open that, it takes a little bit too long. But what I want to show you is this part of the image. So I remind you that when we were talking about glycolysis, so this arrow, it's symbolically shows you the glycolysis path. So you start with glucose and you get 2 pyruvate molecules. Why 2 pyruvate molecules, who will tell me? It was just yesterday, guys. How many carbon atoms in the glucose? Because the second step, well, let's say second part of the glycolysis, because there are many steps, something like 10 steps. But yes, at some point your glucose is 6 carbon atom. So at some point of the glycolysis, you transform that into 2 3 carbon atoms, so 3 plus 3, and then each of them is transformed into 1 pyruvate, because pyruvate, you see, how many carbon atoms? 1, 2, 3. Good. What else? And during this process we generate 2 NADH from 2 NAD+. So this is some sort of electron carrier. I guess we will get into some details a little bit later. But now you see that we generate NADH from NAD+, and you might think that we need a way to regenerate somehow that NAD+ that was consumed during this process. So everything gets down to the pyruvate, we get some ATP molecules, that's great, but we exchange NAD+ for NADH, and now we need some sort of supply of that NAD+. And this mechanism, as you might see, is generating you NAD+ when you are creating this lactate. So that's basically the reason why do we need this process, because this lactate, we don't really like that lactate. So when you have some pain in your muscles, when you did something for some prolonged time, or you ran quickly or something like that, it's basically because of that acidification and lots of lactate generated in that muscle. So you don't like that. Then why do you do that? So why are we generating that lactate if we don't like it? Well, the reason is we need to regenerate NAD+, because otherwise the glycolysis pathway will stuck and we will be not able to obtain some more energy. And that, of course, is no good. We need that energy to move, basically. Here is the example of the reaction. You see the mechanism looks like it goes in 2 steps. I don't think I'll be going through all the details. You probably, once again, you see these histidines that are trying to abstract hydrogens. So you have seen that yesterday. So I will just omit this part. Of course, this enzyme has different forms, so that's a small reminder for you down here. I guess you can look that a bit later. Good. Aerobic condition. Well, citric acid cycle reactions, they occur in the mitochondrial matrix. and cytoplasmic pyruvate and NAD+ must be transported from the cytoplasm into the matrix. Additionally, some enzymes in the citric acid cycle have both cytoplasmic and mitochondrial variants. And so we will, I guess, get to some sort of transport. How do we transport things here and there? But first, the pyruvate dehydrogenase complex. So, what do we want? What do we need? And what is this nice-looking thing doing? I guess I should have some sort of mechanism here. So, what do we want to do? Here we have our pyruvate. Now we want to transform this pyruvate into the acetyl-CoA. That acetyl-CoA will be used later in the Krebs cycle. This nice-looking pyruvate dehydrogenase complex, or PDC, let me show you once again how nicely that looks like. Quite a large complex. So its weight is almost 4 million, with at least 16 chains of 3 different enzymes catalyzing part of the reaction. So this is enzyme complex. So it contains few different enzymes, but these enzymes are working in a concert to create some final result. And in this case, they are called E1, that's pyruvate dehydrogenase, E2, dihydrolipoamide dehydrogenase, and E3, dihydrolipoamide dehydrogenase once more. The molecular weight of the bovine complex is almost 8 million. So it's a really, really large complex. Okay, so returning back. So here are these sort of enzymes. So E1, E2, and E3. I will show you a small video later how that looks like. It's a really nice video, but I just want to go through the mechanism just for a few moments. This thing looks like a hand. So it, at some point, it grabs the molecule that we are performing that reactions and transports from from E1 to E3. Let's look how that should work, and then we get to the video, I guess. Yes. So here is pyruvate. Here is the TPP. It's helping us to perform all these reactions. We are basically releasing CO2, and we get this sort of molecule. So it is covalently now this what is left from the pyruvate is covalently bound to this complex. This hand, so this thing, is basically performing one more reaction, taking away this part. So some covalent bonds are broken, and this part is taken away, and moves it to perform a reaction with creating acetyl-CoA. And then FAD is used to restore the state of this hand. So when reaction is performed, you see it's broken. So this sort of bond that we had here, so here we had a bond between these sulfur atoms, now it's broken. And we should restore that with using FAD. That is covalently bound to the E3 enzyme. And then E3 enzyme restores the state of FADH2 to FAD using NAD+. I guess I have this reaction in a little bit more details. Just one moment. So TPP, or thiamine pyrophosphate, it comes from the vitamin B1, thiamine. And here you see that we have here a carbon atom, so a hydrogen is abstracted, and you see this electron pair is attacking, so that's a nucleophilic attack on this carbon. I hope you remember all these sort of reactions when we are attacking that C double bond O thing, and here we see that oxygen can be protonated. So here is what you get as the result. So a new covalent bond was created, and here is our new molecule. Now we have a minus charge here, so these electrons are used to create double bond here. Here you have now CO2. That's a really good living group, so that gas just leaves. It's the step. So here you see CO2 living. Now what do we have? Once again, we have some change in structure, some sort of isomerization, when these non-bonding electron pair recreates double bond here, and this double bond is basically destroyed and electrons are moved to the carbon atom. Carbon atom acquires negative charge and attacks this sort of hand from the E2 enzyme. Now it is attached to the E2 enzyme. One of the hydrogens is abstracted by the Bronsted-Lowry base. Once again, you see that some reactions are going, and at the end of the day, you have this molecule fully transferred to this sort of hand on the E2 enzyme. Now, what do we need to do? Now we have the CoA molecule. CoA is some sort of a protein, so you have one of the residues that has SH. I guess it's cytosine, if I don't mess up. So I guess this cytosine has SH. This non-bonding electron pair is once again performing a nucleophilic attack. Everything is going. So here you get your molecule. So this is acetyl. Why acetyl? Because this is acetyl group, acetyl CoA, and this molecule will be used in the Krebs cycle. So I'll show you that a bit later. And here you have just few steps that restore the things as they were before we started reaction. So you see that these here we have no bond, so this bond was broken. So here was this bond, and we broke that bond. And now we are using FAD that is covalently attached to E3. That's sort of vitamin B2 riboflavin, and we restore the state of our hand. And now we use NAD plus to restore the state of the covalently bound FAD. That's good. So now let me show you the video, and I hope now you'll understand what is going on. Okay, I guess I'll speed up a little bit.
The product of glycolysis. The pyruvate molecule is the source of carbon and electrons for aerobic respiration reactions inside your mitochondria. Pyruvate enters pores on the mitochondria membrane and is transported into the interior. The reactions linking pyruvate with aerobic respiration are performed by a huge protein complex made with multiple copies of 3 types of enzyme.
So here you see our complex, and these things, these blue things that are moving, that's what kind of enzyme is this? E1, 2, or 3? No, it's not E1. Which one of them did I call a hand? You remember that ring with 2 sulfur atoms? E2. Yes, I called E2 the hands. So these are that sort of hands. It's E2.
The first enzyme catalyzes decarboxylation, transforming 3-carbon pyruvate into a 2-carbon acetyl group, generating carbon dioxide as waste. The acetyl group is then grabbed by the second enzyme, using flexible arms to efficiently transfer the product between reaction sites. In the active site of the second enzyme, the acetyl group is attached to coenzyme A, generating acetyl-CoA, fuel for the citric acid cycle. 2 electrons from pyruvate oxidation are retained by the second enzyme, before passing them to coenzyme NADH, catalyzed by the third enzyme. Coenzyme NADH travels through the matrix, delivering electrons for the electron transport chain. In a sequence of reaction steps, the pyruvate dehydrogenase enzyme complex generates fuel for aerobic respiration reactions of the citric acid cycle and the electron transport chain.
Паш, тебя слышно на фоне, когда ты жуешь?
Да.
Okay. So now let's get to the Krebs cycle. So you saw the pyruvate complex, you saw how that works, and now I guess we have to deal with the main, let's say, thing with the Krebs cycle. I think I will, I don't know, I will cut some, make some shortcuts here and there, because we have, I guess we have too much material to cover, but anyway. So here's basically the Krebs cycle. Here you see the acetyl-CoA that enters the Krebs cycle, and then citrate synthase basically performs, releases that coenzyme A and attaches this acetyl group to the oxaloacetate by generating basically a citrate. So here we have it. So before we had 1, 2, 3, 4 carbon atoms. Now we are adding 2 more carbon atoms. So we have 1, 2, 3, 4, 5, and 6. So 6 carbon atoms. Then as we go through this pathway, we will eventually lose CO2. So here is one step. So we are losing 1 carbon atom. Here's another step. We are losing 1 more carbon atom. So basically we do certain transformations to make, let's say, easier to reject that sort of carbon dioxide, and after we have rejected that, we perform some more reactions to get back to the molecules we have started with, so like this oxaloacetate. So here it is, and here you see some smaller inset in this image that when we get from the glucose to the pyruvate and to the acetyl-CoA, first of all we can get to acetyl-CoA, but by a few different pathways, like we can get that there from fatty acids, I guess beta oxidation, or you can get there from ketogenic amino acids. And from the acetyl-CoA, you can once again go in different directions, like perform oxidation in the Krebs cycle, or you can synthesize fatty acids, or you can synthesize cholesterol and the result steroid hormones and so on, or you can synthesize ketone bodies. There is no way from acetyl-CoA to the glucose, so if we are short on the glucose, and we have a really big problem because our brain uses almost exclusively glucose. What do? We synthesize ketone bodies from the acetyl-CoA. It's not really good, but that works, and we can somehow prolong the time that we can work without consuming some food and some other sugars. Okay. So this is the first, this is the final common oxidative pathway that oxidizes CoA, acetyl CoA to CO2. So we bring two carbon atoms, we reject two molecules of CO2. So we reject two carbon atoms. This is the source of reduced coenzymes that provide the substrate for the respiratory chain. It acts as a link between catabolic and anabolic pathways. It provides precursors for synthesis of amino acids and nucleotides. So all these molecules that we have, all these intermediates in the Krebs cycle, they are used in different places. So it's not, they are not exclusive to the Krebs cycle. It's not like isolated somewhere and that's all. No, they are used all over the place. I guess I have some slide here that shows connection between different pathways, and you will see that they are used basically everywhere. And it is tightly controlled by all these, not all, some of these enzymes I will show you are tightly controlled to maintain the level of these chemical compounds. Good. What's interesting here? We have synthesized two molecules of CO2. So if you consider all these careful enough, where do these oxygen atoms in the CO2 come from? Well, not really, but let's say so. Sugar already contains some oxygen, so we have some oxygen already here that will be used. And yes, we are using some water, so for example here, that contains oxygen that can be rejected as well. What is interesting here that these oxygens in the CO2, they do not come from the air. So that's what's interesting. And why do we use oxygen from the air? We will see a little bit later. Good. Here is some sort of estimate how many of ATPs can we get from the Krebs acid from the Krebs cycle. Well, it's not so easy to calculate that because many old books they perform calculation that gives you 38 ADP molecules. Some more recent estimate give you 32 ADP molecules. That basically depends on how do you take into account all these NADHs that we will later use in the electron transport chain. So we'll get to that. I will show you that electron transport chain, and maybe you'll get some idea why it's so complicated to estimate this sort of thing. Here is example. So what I told you that the Krebs cycle is not isolated, so all the intermediates are used in some different pathways. So it's some sort of on the crossroad of many pathways. And here is the example of the efflux of all the intermediates. For example, alpha ketoglutaric acid can be used to synthesize glutamic acid, and then GABA. Probably you know what is GABA, or gamma aminobutyric acid. Did you ever study neurobiology? I guess not. So this is one of the neurotransmitters. Yes, mediator or neurotransmitter, one of the main neurotransmitters in our brain. So I guess we will get to some of the topic because I really like neurobiology and neurobiochemistry, and we will definitely meet GABA there. Succinyl CoA used to for synthesis of heme or for ketolysis, so many different pathways. Malate can be used for gluconeogenesis. Oxaloacetate may be used to generate aspartate, or acetyl-CoA can be used to synthesize fatty acids. We need them as well as a sort of storage and basically for our cell membranes, so many places we will need fatty acids. Good. And here is, of course, they are not just taken away from the cycle, they can be invested into the cycle. So here are some of the pathways that can replenish that sort of compounds. For example, our glycolysis, so a classical way to get pyruvate that is transformed into acetyl-CoA and then gets into the Krebs cycle. There are some ways to get oxaloacetate and to get into the Krebs cycle. Some amino acids like alanine can be transformed into the pyruvate, and tryptophan can be transformed into alanine that is transformed into pyruvate. We can get oxaloacetate from aspartate. We can use phenylalanine or tyrosine to get fumarate, that is once again a part of the Krebs cycle. We can use even more of these amino acids like valine, isoleucine, methionine to generate propionyl-CoA that is generated with odd chain fatty acids. Well, it's a interesting process. Let's say that if you have odd chain fatty acid, our enzymes are performing all the reactions on fatty acids by manipulating 2 carbons at a time. So when you have an odd chain fatty acid, you do all that manipulations, and at the end you are left with the molecule that contains 3 carbon atoms. And these 3 carbon atoms, they basically require some separate pathway to do something with them, because all the enzymes that did probably all the previous work with the large fatty acids, they cannot do anything because they just work with even number of carbon atoms. Good, and you can get alpha ketoglutarate from glutamate and even more of the amino acids. So there are different ways to replenish compounds of the Krebs cycle and different ways that you can extract them and use for creating something useful. Here is one more scheme that shows you regulation of the metabolite flow from the pyruvate dehydrogenase complex through the citric acid cycle in mammals. So here you see, well, if you look at these steps, everything that is red, that's basically an inhibition, and green, it's activation. And you see there is certain logic to all of these. So if you have a lot of ATP or acetyl-CoA or NADH or fatty acids, it means that you already have all the energy that you need. You cannot use that energy for some reason, so you don't need it so much. And they inhibit the pyruvate dehydrogenase complex, because why do we need to synthesize more ATP if we haven't consumed what we have? On the contrary, the AMP is basically signalizing you that we are in a low energy state, we need energy right now and immediately, and of course that activates the pyruvate dehydrogenase complex. And you see this sort of regulation all over the cycle. So some molecules that may signify some low energy state, they will activate Krebs cycle. Some different molecules that that may tell you that we are in high energy state, that we have enough ATP to perform whatever we do, they will inhibit some of these enzymes and try to make Krebs cycle slower. Good. So here are some details. I guess I will skip them almost entirely. So these are once again reactions as we have considered with the glycolysis. Reaction number 1, citrate synthase takes oxaloacetate and creates citrate. So it's basically taking this part of the molecule and putting it onto the oxaloacetate and transforms 4-carbon chemical into 6-carbon chemical. So here is some nice animation. It changes its conformation on the binding with all that substrates, so I'm not getting into the details of the mechanism, so just a neat picture for you. So here is the mechanism. So 4-carbon oxaloacetate condenses with 2-carbon acetyl-CoA to form 6-carbon compound, the citrate. The hydrolysis of the thioester bond in acetyl-CoA drives the reaction forward. This is an irreversible step, so you use the energy of that bond to, well, basically create a new chemical. So that's what I told you at the beginning of our lectures. We are in biological systems, all the enzymes are trying somehow to couple reactions to get negative delta G at the end, and to make them spontaneous. And for this purpose, they can couple some reaction with very low delta G with some different reaction that we want, but it does not have. So low delta G, it has some positive delta G, but we want it negative. So these reactions are coupled, and you see how that works. Good. One more reaction performed with aconitase. The chemistry of this reaction, so the chemical rationale for this reaction is to move an OH to beta position, which is a subsequent step in converted to a beta CO, so it can act as an electron sink to facilitate decarboxylation in a following reaction. So we will see that following reaction and and so on. So reaction is readily reversible, so you see that delta G naught is not so high, and so on. So here is an example of that aconitase. It performs everything in 2 steps. The enzyme has an inorganic iron 4 sulfur 4 cluster. So here you see that that cluster. I guess it looks really nice. So here is the cluster itself in the form of cubane, so it's some sort of cube. And of course it is stabilized with some cysteines from from the protein, and here you see how that reacts with the isocitrate. The cis-aconitate intermediate in the interconversion of citrate and isocitrate undergoes a 180-degree flip around the double bond, but mechanism is still unknown. So if you are interested, what do we still not know? So here we have a reaction, here we have some sort of where is it? A tentative mechanism, but we still don't know all the details of what is happening there. And we have this sort of nice cluster. Good, what else? Isocitrate dehydrogenase. You see negative delta G naught, that's good. The chemical rationale is pretty clear, so we are rejecting CO2. So we have basically removed one of the carbon atoms, and we are getting some NADH from this reaction. So you will see that whatever we get from the Krebs cycle is always this sort of NADH. Then later we will use this NADH to get some ATP. Good. There are different forms for probably many of these enzymes, so I'm not getting into that sort of details. Here is the tentative mechanism. Once again, nice 3D pictures. If you like, you can consider that in more details. I'm not getting into that. Okay, so fourth reaction, alpha ketoglutarate dehydrogenase performs this sort of reaction. Once more CO2 is rejected. That's in oxidative decarboxylation of alpha ketoglutarate, and basically performed almost the same way. Fifth reaction, succinyl-CoA synthase. Here you see this sort of reaction. Interestingly, it gets from GDP to GTP, so we even gain a little bit of energy during this reaction. Well, GTP energetically is more or less equivalent to the ATP. That's good. And there are certain reactions, I guess we will get to them maybe at some point, that even use GTP as their main source of energy instead of ATP. But that will be sometimes later. Good. And this is the first step in which the energy change in the cycle is explicitly captured in the form of high energy molecule. So previously we had that, but implicitly in the form of NADH. That should be a little bit more processed. Here we have explicitly GTP that can be readily used. Good. Here are once again, some nice pictures for you, and even some links. You can use them to see 3D. Here is the tentative mechanism that we have. Once again, what is this sort of thing? Look at this arrow. What kind of reaction is this? So that's an attack, but what kind of attack? Well, it's not SN2, but yeah, you are close. So this is a nucleophilic attack with the formation of that sort of tetrahedral intermediate. Well, not all reactions are SN1 or SN2, but you remember the, I guess, fourth lecture, we have considered reactions with C double bond R, so with this sort of group. And I told you that carbon atom is a little bit positive, so whatever attacks this carbon atom, well, in this case it's negatively charged oxygen, so this is a nucleophile, and it basically performs a nucleophilic attack, and you get that sort of tetrahedral intermediate and so on. Okay. One more step. This is yet another step in closing the cycle to reform oxaloacetate. So we have already gained a little bit of energy in form of GTP, and we have gained some sort of NADH that we can use later to get energy in form of ATP. Now we should return somehow. No, it's not. We will get to that just in a moment, and we should get back to where we started, because this is a cycle, so we should somehow reform the initial chemical. So here is one more step that goes that direction. It creates us FADH2. No, not bad as well. Here is even one more step. So we are just trying to reform what we had at the beginning. I will skip all the details probably. Yes, I will skip all these details. Here is a video for you. I guess we have no time to look at right now, but that's about the Krebs cycle, so I advise you to look that after the lecture. So now so now you see the last step was the formation of the oxaloacetate, and we have basically closed our cycle. So here, if I return to this point, so we have 8 steps, and the 8th step closes the cycle. So now this oxaloacetate can be once more reused in the cycle. So once more we use acetyl-CoA and go round and round and round. Good. I guess this needs some time to meditate. But let's get to the final part of all of these. So we have already obtained a little bit of ADP during glycolysis. We have obtained one GTP during Krebs cycle, but now we have a lot of NADH, and we will use this NADH to get more ATP. So where are we going? What are we doing? This is a mitochondria. So you see how that looks like. It's some sort of 3D view. It has two membranes, so outer membrane and inner membrane. And here you see this sort of image. And here is an example that shows you where are these reactions performed. So you see some names here. So mitochondria are pretty autonomous, so they have even a little bit of their own DNA. You probably know this DNA is pretty small. It encodes something like 13 proteins, 22 tRNAs, and 2 rRNAs, all for oxidative phosphorylation. So we are now getting to oxidative phosphorylation. Most mitochondrial proteins are encoded by nuclear DNA and are imported. They are inherited only maternally. So the mitochondria that, well, at least I guess 1 or 2 mitochondria is present in sperm, probably 1. But they are typically degraded after fertilization, so they are inherited strongly maternally. That's why we can trace, using the mitochondrial DNA, we can trace back using just, let's say, one part of our inheritance tree. So all people, it turns out that after analysis of lots and lots of mitochondrial DNA and knowing how quickly do we get some sort of mutations in the DNA, we can estimate when lived one woman that we are probably that is probably our recent common maternal ancestor, and there is some sort of estimate. Of course, it may happen that at some point we find some people that have a really different mitochondrial DNA, and we should somehow update these sort of estimates. But anyway, and you probably know the theory of the endosymbiotic origin, so that mitochondria descended from an alpha proteobacterium that was engulfed with the proto-eukaryote around 1.5 or 2 billion years ago. And mitochondria are basically capable of undergoing fission, so they can split. One mitochondria can split into two, and they can fuse. What's even more strange, but yeah, they can fuse, and that fusion, it helps to mix content and complements some sort of damaged mitochondria. Well, damage in mitochondria, that's really bad. So if you have some sort of damage, that may be a really bad disease. And whatever you have in the mitochondria is involved in apoptosis, in calcium buffering, in heat generation. It's a source of reactive oxygen species, and of course the mutations in mitochondrial DNA, they can cause bad diseases like Leber's hereditary optic neuropathy, or lethal infantile mitochondria ophthalmoplegia, or myoclonic epilepsy, and so on and so forth. And have you ever heard news about the kid of 3 parents? I guess that was sort of in England. No? There was at least one case when parents had some sort of defective mitochondria, and they knew that their child will inherit that sort of mitochondria and get some sort of that optic neuropathy that's really bad. and I guess there was at least one case when doctors were able to let's say create a child with the with external fertilization, taking genetic material from three parents. So nuclear DNA was from parents that wanted this child, and the mitochondria were from some different woman that was used as the donor in this case. I'm not sure how it's going, but I know that there was at least one case, and probably they were able to fix this sort of problem with mitochondria. So yes, good. Well, one more tricky question for you: if we can trace some sort of maternal inheritance using mitochondria, can we use something for tracing, let's say, paternal line? If yes, then what? If no, then why? Yes, Y chromosome, absolutely correct. So we get mitochondria exclusively from our mother, so we can trace using chromosomes the maternal line. But when we get Y chromosome, that's exclusively from father, so we can trace that back. Good. Here's once again mitochondria. You see here the outer membrane, inner membrane, the cristae, so it's how they look like, and the matrix. Good. Now we are getting to the process of generating ATP from what we have from NADH. So this is a sort of electron transport. It's called electron transport chain. This electron transport chain is on the inner inner membrane of the mitochondria, and we will see that when this when it performs its work, it's sort of pumping protons in the intermembrane space of the mitochondria. So we will see that. So basically here is some sort of an energy diagram. So electrons are transferred from NADH to the oxygen through a chain of three large protein complexes. So they flow within these transmembrane complexes. This flow is highly exergonic, and it powers the transport of protons across the inner mitochondrial membrane. A fourth large protein complex contains the succinate dehydrogenase that generates FADH2 in the citric acid cycle. So electrons from this FADH2 enter the electron transport chain at Q cytochrome C oxidoreductase. So here you see how do electrons flow. So here is complex 1. Here comes NADH. Some electrons are transferred. Here comes FADH2 and succinate Q reductase. Some more electrons are entering our electron transport chain. It is transported to complex 3, then through the cytochrome to complex 4, and so on and so forth. Let me find you some some more details. Let me show you this one first, how that looks like, and then we get back to our calculations, and that will be good. So here you see how all these complexes are situated. So this is the inner mitochondrial membrane. So you see this is the membrane. Here is the matrix site. Here is the intermembrane space. So that means you have a second membrane somewhere up here, and you are pumping your protons into the intermembrane space. What's happening here? So here you see NADH plus H plus coming here and NAD plus generate. Here you see succinate to fumarate transformation and that's all. So these are chemicals that come here. Electrons, you see the path of the electron, how it is transported through these complexes, and they are passed from one complex to another until basically they finish up in this complex that transforms two protons and an oxygen into water. So we are generating water. This is the oxygen that we breathe from the air. So here it is. So the oxygen that we breathe goes into the generation of water. So the carbon dioxide that we breathe out does not contain any oxygen atoms that we have that we breathe in, because all that atoms they go into the generation of water in the electron transport chain during the oxidative phosphorylation. So here is this summary of the electrons flow and the protons through the four complexes of the respiratory chain. And one more name of the electron transport chain. Electrons reach ubiquinon through complexes 1 and 2 as well as through several other paths. Reduced Q serves as a mobile carrier of electrons and protons. So here you see that ubiquinon can transfer basically electrons from one complex to another. Just takes them and diffuses and then performs the reaction. During getting these electrons, so you see that electrons are passed from one center to another, it loses some energy, and this energy is used to pump some protons. So you see 4 protons are pumped from the internal space into the intermembrane space. Here are different 3D views of these molecules. I'm afraid we don't have enough time to get through all of these. This is, for example, one of the pumps. So this is complex 1, and here you see one of the pumps that takes protons from the internal space and pump them into the intermembrane space. So here as an example, you see that when the conformation changes, you see these residues, that's NH3, and when it is in the open conformation, they carry some positive charge, so this is NH3 plus, and that thing had NH3 plus, but they are more close together, and this destabilizes that plus charge, so one of the protons is more willing to leave, so it has a lower pKa, so it wants to go away. In this sort of conformation, this positive charge is stabilized and in some sort, let's say, neutralized with 2 oxygen atoms. And here, so these are 2 oxygen atoms, so some hydrogen bonds may be created, some negative charge, they may have some negative charge, so this is stabilized and it's not influencing so much this residue, and it still wants to retain its protons. So that's basically the way how this pump takes protons from one place and put them into another place. So pKa is drastically changed, and in one conformation they want to leave, and in another conformation they do not want to leave so much. Here are different examples of these complexes and how electrons are transferred in each of the complexes. I guess there's no need to get into all of the details. Just you see that ubiquinon H2, so 2 electrons from ubiquinon H2, they take different paths in complex 3. So 1 electron moves to risky cluster, so there is once again iron and sulfur, and the other to the cytochrome Bl. So the electrons move to the risky center, then move to the cytochrome C1, and then to the mobile electron carrier cytochrome C, which is bound to the complex in the intermembrane space. Here are some more and more structures I guess I will omit that all. I will show you just what just these sort of problems, and then maybe one small video, and that will be great. So you see in these sort of diagrams, when I show you all these free energy, so we are somehow calculating this sort of free energy and how electrons are moving, so on and so forth. But when you get to some real life data, you will see that something a little bit different is reported. So some values like E are reported. They are called potentials or cell potentials. Or even you will see a half cell potential. And right now I want to show you how they are obtained and how do we perform some calculations with these sort of potentials. So you remember that we measure basically everything with the change in free energy, delta G. That's a measure of the maximum amount of work that can be performed during a chemical process. That's pretty simple. If we can attribute this work to a process of moving charges, well, basically sounds pretty reasonable. What is chemistry? Chemistry is basically moving charges, most often electrons. So we can define some virtual potential and rewrite our free energy in this form. So what that means? If you remember something from, I don't know, from electrostatics or whatever, if you have studied some physics, you know that you can calculate work performed on a charge or by a charge if you have some electric field, like result of the multiplication of this charge on the field you have moved this charge in. So here is basically the formula. So D is the charge. It can be -1 for electron or +1 for proton. F is the Faraday's constant. So that's basically the charge of 1 mole of electrons with the minus sign. So we won't always estimate in chemistry everything in moles, so that's just some sort of convenience. And E is our potential of the cell. And here I rewrite our formulae once again. You remember that delta G is basically delta G naught plus RT logarithm Q, so that's what we have. These delta G naught we can treat as well as ZF and some E cell. And everything here we can treat as the ZF E cell. So this is E cell naught for standard conditions, and this is some sort of E cell. In case of the equilibrium, we set delta G to 0, and we have this formula. And from this formula we can get E cell naught. So here how that looks like, and that's why you can have some sort of connection between the equilibrium constant and E cell naught. Now what do we how do we calculate anything? So E cell naught can be thought as E naught of the final state minus E naught of the initial state. So electron is moved basically between two states. We have different field, and we can calculate some sort of gain or loss in energy. And that's exactly what a voltmeter will show you if connected. So E's of states are called potentials of a half reactions and are measured with respect to the standard hydrogen electrode. So you can find them in tables. So you will find some data on that. For electrons, initial state is anode. Here happens oxidation, and oxidation is a loss of an electron. So electrons are lost on the anode. Final state, cathode. Here is performed reduction, and reduction is accepting of an electron. So values of E naught for half reactions cannot be added to give the sum of the half reactions. That's intensive property like temperature. So you cannot combine two glasses of water with 50 degrees C and 50 degrees C together to get 100 degrees C. That's impossible. So this is intensive property, so we remember that. But values of delta G can be added. That's extensive property. And I will show you an example of calculation. So here is an example. So we try to estimate, is the following reaction spontaneous? We measure that by checking the sign of the delta G. Now we need to get delta G, but we don't have that for half reactions, but we have these sort of E naughts. So I have two half reactions. I have the final reaction that I want. I hope you remember how you did the problems with the enthalpy. Absolutely the same thing. So I try to combine these two half reactions to get my final reaction. So how do I do that? I set the direction for one reaction. You see these arrows are both ways. You do not change sign of E naught when you change the direction, and when you multiply by something you do not. That's an intensive property. This reaction, I swap something, I change somehow the direction, I choose the direction, I multiply by 3 to get these coefficients. If I add everything together, I will get what I need. But now I need to calculate how that works. So here you see E naught here and here are basically the same. They did not change. But now I calculate on the cathode the delta G naught on the cathode. That's minus 6F times E naught. So 136 as its E naught. Where did I get this 6? That's the number of electrons transferred in one reaction. So for each mole of something reacting, I transfer 6 electrons, so it gets 6 times bigger. And here I can get this delta G. For this reaction, once again I get delta G as minus 6F times E naught. That's what I get. What now? Now if I get cathode minus anode, so at this step I perform, let's say, understanding who rejects electrons and who gets electrons. So one reaction, this reaction rejects electrons, this reaction gets electrons. I mean the other way around. So this reaction accepts 6 electrons, and this reaction donates 6 electrons. So you see the way it goes, it generates 6 electrons that should go somewhere else. And this reaction should accept 6 electrons to proceed. So this is cathode, this is anode. And now if I take cathode minus anode, I will get my final result. So here is my final delta G. You see, I could have performed the same thing by taking these E naught for cathode minus E naught for the anode, and then multiplying by 6F, because I transfer 6 electrons. And the same way I can just calculate delta G for each of them, and calculate the overall delta G by taking one minus another. So that's the same thing. Here is some other example for the lead acid battery. I hope that you have seen them. So this is cathode, anode, and the overall for E cell you get 2 volts basically. Of course you can say, well, these batteries are mostly 12 volts, but if you look inside you see here 6 compartments, and each compartment generates 2 volts. So being correctly combined in a series, I guess, you will get 12 volts overall. So that's what we get. Okay, I guess I'm almost I have almost no time, but I have almost finished. The last part, we have a lot of hydrogen atoms in the intermembrane space. What do we do with them? We generate ATP with the ATP synthase. What does the ATP synthase? This is this nice looking thing. I'll show you a video. I just want you first tell you the thing. Here is the scheme for a typical enzyme. So here we have free energy, here we have enzyme plus substrate, here we have enzyme plus product. And here is this enzyme helps to overcome this sort of transition state. What does the ATPase? It basically moves all the problems to the problem of the mechanical dissociation of the product from the enzyme. So here you see I have enzyme with substrate, really small peak, really small transition state. I get them bound together, so enzyme and substrate are bound. Now one more small transition state and I get product. So reaction was performed, I get product, but this product is tightly bound to the enzyme. And on the decoupling on the product from the enzyme, that's the place with the highest transition state. So here is the problem. So in typical reactions, you are trying to solve the problem just by making this barrier lower, this potential barrier lower, but in this case you are moving the problem to the mechanical decoupling of the products from the enzyme. And I will show you that basically these ATPase, here how looks the catalytic site. It has few different conformations like O state, L state, and T state, and when it binds in the L state, basically substrate, so ADP and a phosphate, then it closes, reaction is performed, and then the problem is to throw away whatever has reacted because it's tightly bound to the enzyme. And I will show you how that works. I guess the last minutes the best way will to look the video and get the idea how that works.
We are looking at the turning gamma subunit and the epsilon subunits, and there's the C ring in the membrane. The F1 complex is outside of the membrane, while the F0 complex is embedded in the membrane. There's the stalk, including the delta and the B subunit.
So here you see the I guess the catalytic part, and here you see how that motor is rotating. I guess
the alpha subunits are shown in light pink. The beta subunits are magenta. In the middle, the central stalk is rotating. The B subunit stabilizes the hexamer, which means that the hexamer can't rotate. We'll now look at a rendering of known structural states and the conformational changes that are happening. The beta subunits are seen as a ribbon diagram. The ADP and inorganic phosphate diffuse into the active site, and the ATP has just been released, and simultaneously the conformational change occurs in the beta subunit. If we zoom into the active site, we can see the molecular structure of the substrates and the chemical reaction.
So here you see ADP bound, and we need one more phosphate to be attached to get the ATP.
Here is ADP with its 2 phosphates. They are all held in place by a network of hydrogen bonds mediated by the amino acids in the folded structure of the beta subunit. Hydrogen bonds stabilize the base and the sugar, but really primarily the base. There's also a magnesium ion that plays a critical role in not just stabilizing, but also neutralizing the charge of the phosphates. Notice the conformational changes that are happening when the inorganic phosphate comes in. There is the promotion of a planar transition state. Synthesis of a new phosphodiester bond is catalyzed.
So here you see it's basically a transfer of inorganic phosphate is performed, and from the ADP you are now getting ATP.
And then the subunit switches to the open conformation. Upon this reconfiguration in the beta subunit, ATP is ejected from the active site. The enzyme then resets.
Well, the moment of the rejection, that's the more crucial thing, because that's the place when we spend, let's say, most of the energy that we need to synthesize the ATP.
Restarts the synthesis of another ATP molecule. This is a summary of the events at the active site. It is also intriguing just how the C ring rotates. We can start with the analogy of a water wheel. We can easily grasp the idea of how a water wheel rotates. It's driven by the flow of water, allowing the wooden fins to go in a particular direction. Thus, the rotation happens in a specific direction. If the water was to flow in the opposite direction, you would see rotation of the water wheel in the opposite.
Here you see protons in the intermembrane space, so that's between the two membranes, and they are getting back into through this inner membrane, but when getting back, they are rotating all these proteins, and it's really quickly rotating. I guess
Direction.
something
Here we see the C ring and the A subunit. We don't know the full structure of the A subunit, but we know that there's an access channel for protons on the A subunit right in the intermembrane space, and there's a separate egress channel for protons on the matrix side. In this animation, you see protons flowing out from an interface between the C ring and the A subunit. But what drives the specific direction of this rotation? For example, if we were looking down, it is always a counterclockwise rotation. Why and what drives that? On each of the C subunits within the C ring, there's a proton binding site. Notice the high concentration of protons on the intermembrane side and their flow to the lower concentration on the matrix side. Protons load from the highly concentrated intermembrane side onto the C subunits, and they are released on the matrix side where the concentration of the protons is low. But why can't protons go around the other way as well? In principle, just as in the water wheel analogy, the C ring should be able to rotate in either direction, shouldn't it? There are two interconnected reasons for this unidirectional movement. The amino acid side chains in C subunits would repel the bound proton, so that when a proton loads from the intermembrane side, the C ring cannot rotate back in the opposite direction because it is energetically unfavorable. Rotating counterclockwise, on the other hand, is energetically favorable because there are no repulsive interactions. Remember, there's a concentration gradient that favors binding of protons on the intermembrane side and the release of protons on the matrix side. So in summary, the proton gradient determines the direction of the rotation, favoring movement in the counterclockwise direction of the C ring. This avoids unfavorable interactions with the amino acid side chains of the C ring.
Okay, so I guess I'm not getting into the details of all that subunits. Here is electron transport chain at glance. I guess you can look that on your own. And that's basically everything I wanted to tell you. I have a lecture, I guess, but I hope you liked the pictures, and maybe you will watch videos a bit later. Okay, do we have any questions or comments so far? No. Okay, good. If no comments, no questions, I guess it's a good point to stop. So have a nice evening and see you next week.